Alrighty. Oh yeah, I lived in the Keys for a while when I got out of grad school. I actually got a job down there working in the Keys at a lab on a Long Key, which is just a 15 or so miles up from Marathon. Awesome job. All I do was scuba dive all day long. Can't beat that. Alrighty, folks. So when we were together last time, we had just talked about some of the general characteristics of land plants. Talked a little bit about how those characteristics, how those traits arose and allowed them to survive on land. Of course, we've talked about one of the biggest concerns about living on land is going to be the desiccation issues. So that's something that plants definitely had to conquer before they were able to continue to move out onto land. Initially, plants would have grown near the water, right? Sort of like the green algae you might see on the side of a lake. But as time went on, evolution being the way it is, organisms began to evolve this ability to actually move out into much more drier climates. So we've talked about that. But what I want to do today is I want to go through and start talking about some of the plants. Today we're going to discuss these two groups of plants. The non-vascular bryophytes, and then we'll work our way down here to the vascular plants. We'll get to the seeded plants, which is the subject of the next chapter. So we're going to talk about liverworts today, mosses, hornworts, all the way down to about right here, right, the ferns and everything. Now, notice when you look up here in the red text, what you're seeing there. We got non-vascular plants, and we have vascular plants. And then within that, we've got seedless vascular plants and seed plants. So we see this gradual evolutionary change that's occurring among these plants. Now I'm getting too warm. Among these plants as they begin to move out more and more on the land. Seeds are very effective ways to distribute plants on land. We won't get to those today, but when we do, we'll talk about that a little bit more. But let's get into the bryophytes first. So we're going to be looking at these guys, the bryophytes. So the bryophytes include the liverworts, the mosses, and the hornworts. The bryophytes were probably the first that diverged from a land plant common ancestor. So whatever that first plant was able to survive and move out on the land, we think that probably the mosses and the bryophytes and their relatives. Hi, Daniela. Oh, she's not quite yet. Were probably the first ones that evolved out of that common ancestor. But those first group of plants were seedless, so they had spores, and they were non-vascular. Now, what I mean by vascular is the fact that they had tubes inside of the organism by which they could move nutrients and water around. Think of the vascularization in plants as very similar. Hello, Daniela. As very similar to the vascularization in animals, right? Our blood vessels and that sort of stuff. So these first group of plants that we're going to talk about had no seeds and no vascularization. They were called non-vascular, or another word for that in red here is non-tracheophytes. A trachid is a specialized transport cell, okay? Large plants are going to have trachids. That's the only way they can effectively move nutrients and water around within their bodies, if they have these special transport cells. If you're a bryophyte, if you're a small non-vascular plant, well, I shouldn't have said the word small. If you're a non-vascular plant, you're limited to staying very small. And the reason why you have to be very small is because the only way you can move things in and out of your cells is through diffusion or osmosis. And remember the thing with diffusion and osmosis can only move so far into a multicellular organism, right? So if we had some layers of cells here, a little bit of a delay in my pen. Diffusion and osmosis, say of water. So water could, through osmosis, move into the cells, but after a while, the cell layers get so thick that the cells further down or deep Sure wish I knew why it does that. Seems like just out of nowhere sometimes. Yes, we got. Oh, you and Danielle. Very nice. Alrighty, so like I said, the bryophytes are non-tracheids, and so that's why they generally stay small, right? You don't see really any big mosses. That's because they have to live very low to the ground. They don't have any supporting structures. They don't have the like the supporting structures that other plants do. The prominent cell wall that allows them to stand up against the force of gravity. And they're going to stay small because, again, diffusion osmosis can only work through a certain layer thickness of cells. Beyond that, it's inefficient. So these vascularizations that come a little bit later on are going to take care of that problem, because scattered in amongst the cells are going to be these transport cells that move these materials around. Very simple. The bryophytes are relatively simple organisms, highly adapted to very diverse terrestrial environments, though, which is something you might expect to find in an organism that moves into a new environment, right? Moving out onto land exposed organisms to a whole bunch of different types of environments. And if you had the ability to deal with that, if you had the ability to adapt to that, right, then you would be able to survive, okay? So these very simple, relatively early organisms did a very good job of adapting to these very different types of terrestrial environments. Of course, we talked about what gametophytes and sporophytes are, but among bryophytes, the gametophyte is much larger. It lives longer, it's very conspicuous, and it's green and it photosynthesizes. Where the sporophytes, remember the sporophytes when we went out after lab the other day, the sporophytes were little wispy brown things growing out the top of the gametophytes. And in bryophytes, the sporophytes are very small. They don't live very long. Really, their only job is to live long enough to make spores. It's the gametophyte that does all of the rest of the work, right? The photosynthesis and that sort of stuff. And among bryophytes, the sporophytes are also dependent nutritionally dependent upon the bryophytes. If you remember in class when we were looking at the slides, sporophytes grow right out of the archegonium of the gametophyte. Remember that little vase-like thing that we looked in some of them? There's this little vase. There was an egg down here at the bottom. Sperm makes its way over and fertilizes the egg, and what grows out of that is going to be the sporophyte, that little thing with the capsule where it then produces spores. Now, let me say something about when we use these words dominant and not dominant. It's not the same as when we're talking about in genetics. It's not the same when we're talking about in behavior. Among plants, when we talk about which generation, and this goes back to what I've been talking about with that alternation of generation, right, where you have two multicellular generations. When we're talking about dominance in this context, what we're talking about is the stage that's the most obvious, right? Like in the gametophyte with mosses, the gametophyte, that green that you see in the picture there, that's the most obvious. That's the most conspicuous. So sometimes the term they use is the dominant. So the dominant generation among bryophytes is the gametophyte, all right? It's the bigger one. It's the one that you typically see when you think about a moss. Many of you maybe until yesterday never realized there were those little tiny sporophytes that stuck up out of a moss, but their sporophytes are going to be a lot smaller. Mosses are generally found in moist and shaded environments. They don't do very well in the sun, and they need those moist environments because they need water for sexual reproduction. You see, their sperm are flagellated, right? They have a flagellated sperm, so they can't swim. You can't use a flagella to swim through air. You're going to have liquid. So basically what they do, I showed you guys in lab the other day, is the sperm swims from the antheridia, the male sperm producing part, to the archegonia through a series of droplets of water. So a bunch of little droplets of water, these little sperm kind of make their way through those droplets of water until they finally connect up with an archegonia. That's why mosses are going to need that kind of liquid to survive, right? Very interesting how that works together, how all these organisms always seem to fit into their environment and adapt to it. All right, now, among the bryophytes, we have the liverworts, which I introduced you guys to the other day. And again, the reason why they're called liverworts is because the gametophyte, this is the gametophyte, this leafy part, this is the gametophyte here. The gametophytes are liver-shaped. A phalloid, folks, or a phallus. A phallus is it's an undifferentiated leaf. call it a beginning kind of leaf. Liverworts and bryophytes never really truly develop leaves in the sense we think about on trees. So a thallus would be not a leaf, but a leafy, like I put in quotes here, a leafy kind of thing. Now, of course, photosynthesis happens with the thallus, but again, they don't make leaves the same way we see in other plants. The gametangia, the structures that form the gametes, are umbrella shaped. The female ones look more like a palm tree. You see right there, we looked at those. Remember, we looked at that under the microscope. Recall that the difference between the liverworts and the mosses were that in the case of the liverworts, the archegonia were on the underside of the of the gametangia. Remember, in the other ones, in the mosses, the archegonia was actually facing up. All right, so that was one of the questions in your lab manual. In mosses, the archegonia face up, and in liverworts, the archegonia, the female part, are on the underside of those little palm tree-looking things facing down. And inside of that, of course, you see are the capsules where there are the sporangium. Now, the male ones, the female ones look like the little palm trees, and the male ones are going to be much flatter. This is a male one down here. These are you can see the gemma cups. Remember, liverworts make gemma cups. Gemma cups is where asexual reproduction occurs. The gametangia, the male and the female gametangia are going to produce sperm and egg, and that's sexual reproduction, but you can also grow another liverwort out of a gemma cup, and that would be asexual reproduction. The phylum name of liverworts, I think, is kind of interesting. The phylum name is Hepatophyta. Hepatic. What does the root word hepatic mean? Any ideas? Hepatic is liver. Okay, so liverworts are members of the phylum Hepatophyta. All right, that's one bryophyte. The other bryophyte that we're going to discuss, or one of the other bryophytes that we're going to discuss, are the hornworts. And the hornworts also have an interesting name, Anthoceratophyta. Antho means horn or antler. You can think about the antlers on a deer. Now, these organisms are a little bit different. There's the gametophyte, and there's the sporophyte. Now, the sporophyte looks a little bit differently than it does in the other ones. You remember in the ones we looked at out in the woods, there was this thin little brown stalk, and then there was a capsule at the end where the spores are made. That was the whole sporophyte. This part of the sporophyte, in those ones that we looked at the other day, is called a seta, S-E-T-A. It's the stalk that holds it up. In hornworts, there is no seta. All right, I'm back again. Okay, so there's no seta in the sporophyte of a hornwort. What we mean by that is, although it looks like that's a stalk, that entire structure that you see there is sporangium. So remember, in the other ones, they had the seta, the stalk that held it up, and the sporangium was the thing at the top. This is the sporangium. But in hornworts, the whole thing is the sporophyte. The whole thing is the sporangium. Hornworts are very often what we call pioneer species. Pioneer species is a term we use to refer to organisms that are many times the first organisms to move to an environment. Many times pioneer species tend to do very well in diverse environments. I mentioned that with the bryophytes. So this kind of harkens back to their early evolutionary days, right? Pioneer species would have been really good at dealing with those kinds of marginal environments in which many species of organisms wouldn't be able to survive. And one of the things that allowed these pioneer species to actually survive in some of those extreme environments was the fact that they have a symbiotic relationship with nitrogen-fixing bacteria. Oh yeah, Ace, I've tried. I'm sorry you weren't feeling well. So you're feeling better. That's good. Now is the rain and the nasty weather starts, huh? So those symbiotic nitrogen-fixing bacteria allow, remember, usually in plants, the one essential ingredient that is usually limiting in plants, in other words, whatever is limiting is what is in short supply, is nitrogen. And that wouldn't necessarily be available in the soil. Now, there's plenty of atmospheric nitrogen, but plants can't use atmospheric nitrogen, right? They have to use nitrogen after it's been fixed, after it's been chemically converted. And oftentimes it's chemically converted through the symbiotic nitrogen-fixing bacteria. And hornworts live with the symbiotic bacteria in their tissues and allows them to get access to that nitrogen nutrient that they need to survive. All right, now let me say a couple of things about hornworts so that you don't make a mistake in your lab manual if you do come across a hornwort. There are things that are called hornworts by common name, but they are really not hornworts. These are hornworts, but they're not truly hornworts. This is actually a seeded vascular flowering plant, Ceratophyllum echinatum and Ceratophyllum demersum. Both of these occur in Florida. I've seen some of this growing in some of the ditches alongside the trail or alongside the street. But this is not a true hornwort. So if you look that up in your when you're trying to identify it, be careful when you come across something if they call it a hornwort, because it's not a true hornwort. And then the last member of this group are going to be the mosses, which is in the phylum Bryophyta. Now, I know it's a little bit confusing. We're talking about bryophytes. Bryophytes include the liverworts, the hornworts, and the mosses. The only member of that group that's in the phylum Bryophyta is the mosses, right? And this is obviously a nice picture of sphagnum moss, what we looked at the other day. And there you go. You can see the sporophytes right there. You see what I mean by the fact that they have a setae, they have a stalk, the whole thing is the sporophyte. Down below, of course, is the gametophyte, and then the capsule at the top is where the spores are going to be produced. The gametophyte, you notice, is a real small, it has leaf-like structures, although, again, they aren't really truly leaves. And the gametangia, the antheridia, or the archegonia, that's what we mean by gametangia, are going to form at the tips. This is what you looked at underneath the microscope. This is the one where the archegonium was facing up, right? Different than the hornworts where it was facing down. Oh, wait, I'm doing my hands. I don't have my camera on. There we go. So these mosses, the archegonia face up, and in the liverworts, they face down. Sporophytes have stomata, that opening that allow gases to exchange. And of course, when plants moving on the land, that would have been important as well, right? To bring in carbon dioxide and get rid of oxygen. The sporophytes that we saw the other day, the brown ones you see there, are no longer photosynthesizing, but they can photosynthesize until they start producing spores, and then they turn that brown color. Flagellated sperm, like I mentioned, so flagellated sperm are obviously going to need water to survive. All right, so again, using those words, the dominant generation, if we looked at a moss, the dominant generation is the gametophyte, right? We see mostly gametophyte here. Sporophytes, which is the other part of that alternation of generation, are usually small and relatively inconspicuous. Okay, and speaking of mosses, everybody remember that this is not a moss, right? Spanish moss in the left is not a moss. It's actually a flowering plant. And then this is called deer moss or reindeer moss. And that's not a plant at all. That's a lichen. And lichens aren't plants. Lichens are a symbiotic relationship between a fungus and some sort of photosynthetic cell. Could be a green algae. Could be a prokaryote. Could be a blue-green algae, which are prokaryotes. Right? And they live in this symbiotic relationship. They live in this symbiotic relationship for so long that people who study lichens actually call these species, even though that really screws up our definition of species from the beginning of the semester, right? When we were discussing about what makes a species, lichens are actually two organisms, in some cases, like with the blue-green algae and a fungus, from two completely different domains, and yet we're considered them one species. They are obligate of symbionts. In other words, they can't live apart from one another. If you were to tease the algal cells out of the fungus would die and the algae would die. They need each other to survive. All right, let's take a look at bryophyte reproduction, and in keeping in what we know about the alternation of generation. Right? Alternation of generation means that you have two multicellular stages. You have one that's haploid, and that's the gametophyte, and then you have one that's diploid, and that's going to be the sporophyte. That's the only diploid part. All that green down below here, that's all haploid. So let's start up here with the dispersal of the spores. So let's start right about here. At the very top of a sporangium is a thing called a peristome. You look down below, you can actually see a microscope shot of a peristome. And what a peristome does is when the peristome opens up, it releases spores out in the environment. Lots of spores. Look at up to 50 million spores. Lots of spores, remember, because most of them aren't going to survive, are they? Now, if you're wondering what those neat little teeth or little projections around the peristome are, what's a good analogy I can use for that? It causes them to spray out. Does that make sense? You know, in other words, instead of the spores coming out in one sort of one grouping, those little teeth that hang over cause it. I'm trying to think of a good analogy of this, like water coming out of the hose. If you just had the water coming out, it'd come out straight. But if you put your finger over it, then you would cause it to spray. That's kind of what those little projections are doing. And the reason why we want to do that, of course, is dispersal. Right? We want to disperse them. Now, back up for just a second. The way those spores were produced was by meiosis. And remember what I've said to you guys, when you see the word meiosis, you should immediately think variability. Two types of cell division you learned about in Bio 1, meiosis and mitosis. Mitosis makes lots, mitosis makes quantity, meiosis makes variability. Now, meiosis may also produce a lot of cells, in this case, a lot of spores, right? Spores are the cells, okay? But the true purpose behind meiosis is to produce a lot of variable spores. And I hope I don't have to go into any discussion about variability, right? We all understand why variability is so important. All right, so these spores make their way out into the environment, and in some species, what we have spores that are bisexual spores. I mentioned the other day about a bisexual gametophyte. Bisexual spores means there are basically male spores, if you will, and female spores. So if we follow the top part of the green arc there, if we're going along in this direction, if the spore finds a place to germinate, right there, then it begins to grow into something called a protonemata. That protonemata continues to grow, gets thicker and thicker, and forms something called a bud, which is only one cell thick. Remember, these guys don't have vascularization. So in order to do diffusion osmosis, they're going to have to remain relatively thin. And in the case of this male spore, then eventually grows into the antheridia, right, the part that produces sperm. Now, these pictures look familiar, right? This is what we looked at under the microscope the other day. There's the antheridia. Those are the sperm. And on the other side of the situation, right, we have what's growing, a female gametophyte, and the female gametophyte has the archegonium with the egg. The eggs are not dispersed. Only the sperm is dispersed, swimming through little droplets of water until it finally finds its way to an archegonium, swims down the archegonium, fertilizes the egg within the archegonium, that forms a zygote, and then growing out of that archegonium is going to be a young sporophyte, and eventually that's going to produce a capsule and produce more spores. Now, as we said, among these organisms, it is the gametophyte that is the most dominant, and the sporophyte is less conspicuous, and it is also dependent upon the gametophyte. Notice that the sporophyte grows right out of the middle of the gametophyte. So it actually receives its nutrition, right? It receives its nutritional elements from the gametophyte. That's why we say it's nutritionally dependent. The only purpose, now this is something quite different from animals, the only purpose of producing a zygote, the only purpose of fertilization to produce a zygote is to produce variability, right? Because that zygote is going to grow into another sporophyte, which is going to produce, again, a lot of variable spores. The diploid stage, unlike us, right? Your diploid, in animals, diploid is the most common or the most conspicuous stage. But unlike us, in mosses, the most dominant and the most prominent stage is going to be the gametophyte stage. The sporophyte stage, the diploid part, because sporophytes are diploid, you get that from fertilization, that's only produced for a short-lived period of time, only long enough to produce more of these variable spores. Any questions on that? All right, so that is our, oh, let me point out a couple things before I go on. Rhizoid. A rhizoid is not the same as a true root, okay? Rhizoids anchor the organism to the soil, to the substrate, but they don't have a trachid. They have no tubes inside of them. They have no conducting cells that I've been talking about. So botanists, by definition, a root does two things. It anchors the organism, and it also allows it to absorb water through its conducting cells. Mosses have the anchoring part, but they don't have the conducting cells, so they're called a rhizoid, to differentiate those from a true root. It's amazing, 50 million spores, and that's coming out of one of those sporophytes. So just think about all those sporophytes that we saw in that, we saw when we were out in the woods the other day, as well as, look at all these 50 million spores coming out of each one of those. Life must be rough for those spores, right? Most of them are not going to survive, so you're going to overwhelm them with numbers, just like we've been talking about all along. If you're not going to really take care of your offspring, then make lots of them, because you know most of them aren't going to survive. All right, so that completes the non-vascular plants. Let's move on now to vascular plants, plants that had those conducting tissues. So the bryophytes were the dominant land plants for about 100 million years, and wasn't about 425 million years ago that vascular plants began to arise. The first vascular plant was Cooksonia, or at least one of the first vascular plants. This is a fossil of Cooksonia. Cooksonia is not around anymore. But this is what Cooksonia looked like. Those are sporangia that are at the tip there. This down here, even though it looks like an actual picture, that's a computer-generated picture of what we believe Cooksonia would have looked like. Right? So that's not a photograph of anything. Right? Cooksonia, you see it over on the left-hand side. Cooksonia had rhizoids again, only for anchoring. They also had stomata. These sporophytes in this case were now independent. In other words, they weren't dependent upon the gametophyte for nutrition, but still these organisms were relegated to being very small, usually only a few centimeters tall. They had no true roots. They had the rhizoids, right? But they had no true roots, and they had no leaves. So because of that, that basically limited their ability to get really big. But as we move along, we're going to find out that this trend of vascularization was an important hallmark in these plants because it allowed them to get bigger and taller. You didn't have to use just diffusion and osmosis. So let's look at some of the derived traits of vascular land plants. All right, first of all, the sporophyte is going to become dominant now. So in other words, when you think of a vascular plant, I'm going to come back, folks. When we think of a vascular plant, and vascular plant means it's a trachid. Your windpipe is called your trachea. Trachea means a tube, a pipe, okay? So ferns have trachids. And this is the gametophyte of a fern right there, little tiny thing. Oftentimes, you'll never see a gametophyte. Even if you found ferns out in the wild and you dig down into the soil, the gametophyte is so small and delicate, it usually is destroyed by just the act of you digging into the dirt. What is growing out of that gametophyte right here, this is going to be it's actually going to be the fern itself. The fern frond, it's called a frond, not a leaf, to be precise. The fern frond is the sporophyte. So if you look between those two, we got the bryophytes on the left that we just went over, and now we have the trachids on the right. You can see what I mean by how the dominance has shifted. In the bryophytes, the gametophyte was dominant and conspicuous, whereas in the trachid plants, it's the sporophyte. Remember when I showed you those resurrection ferns, and on the back of them they had those little dots, the sori? Sporophytes produce spores. So the leaf of the fern, of those little resurrection ferns, or the frond of those little resurrection ferns, those little dark spots, the sori, where the spores were being produced. So that's a spore-producing structure. But now that has changed as we've gone from bryophytes to tracheids, right? We move from the sporophyte or the gametophyte being dominant to now the sporophyte is dominant. It's not only dominant, it's also independent. In other words, it doesn't need to be nutritionally dependent upon the gametophyte for survival. Now, speaking of tracheids, the kinds of tracheids that were developed in these land plants were something we call a xylem and a phloem. Those are the conducting cells. So let me show you what they look like. Okay, the xylem are tracheid cells. They're tube-shaped tracheid cells, and they're the ones that move water and minerals up into the plant. They're lignified. Lignin is that really strong, durable organic material that plants use, and they coat the cell walls with these, and it makes it rigid so the plant can stand up. If we were to take a cross section through the stem of a plant, we could see the xylem and the phloem. It's actually labeled here. The xylem is going to be these cells that are near the inside. And if we looked at a microscope a little more closely, we could see that indeed they are tubes. So this is a cross section. What we did is we took the stem and we cut it this way. So those are the tubes. So you're kind of looking down into the plant. You can see it down here as well. So the xylem is responsible for transporting water and minerals up the plant. So the xylem would be what takes the water from the roots up into the rest of the plant and out to the leaves. And the phloem is what transports materials like sugars and amino acids and other metabolic products throughout the plant. Now, in school, I always remembered xylem up, phloem down. So I can remember which one did what. The xylem carries the materials up, and the phloem carries the materials throughout the plant. Now, this was a major change in plants. Having the ability to conduct water, to conduct minerals, conduct metabolic products, sugars, food, all that sort of stuff, having the ability to move that throughout the plant through these conducting cells then mean they could get larger. You remember the thing with the bryophytes is they stayed really small and low to the ground because they were confined to just diffusion and osmosis. Didn't have vascularization. Once we add vascularization, then you can get larger and you get more complex. They can increase in size, they can increase in height, and they begin to outcompete other species and outdisperse them. If you think about it, if you can now grow taller, you can outcompete other species because for another reason, you can shade them out. And then getting taller also allows you to disperse your spores further and your seeds further because you're up higher. You know, if you think about that little moss down there, when they're dispersing their spores, you're kind of limited just a few inches off the ground. But if you think in the xylem and the phloem and some of the most extreme examples, like those really tall trees that grow out in California, things are 300 feet tall. That's a really good way to disperse your reproductive cells, right? You got a huge area by which they can expand out into, and because you're tall enough, you got a better chance of dispersing them out in a wider range. So you see what happens through time and evolution, like we've been talking about, is organisms begin to adapt to their environment. And one of the things that they do is they begin to learn how to compete with one another, and they learn how to engage in much more effective means of dispersal. Xylem and phloem allowed vascular plants to do this. Vascular plants also have true roots, right? So they anchor and they can absorb nutrients and water. Remember, that's the difference between a true root and a rhizoid. Not a rhizome. A rhizome is a horizontal growing root. Okay, that's a rhizome. But a rhizoid, which was the little things off the moss that anchor but don't absorb nutrients. Remember, they're not tracheids. Mosses have no xylem, have no phloem. Roots, we think, probably did evolve from stems, though, underground stems, which would have been a really good example of convergent evolution, right? So we have this idea of these stems, which would normally hold the plant up, evolve through time to become the roots that anchor the plant into the ground. And since the stems would have been vascularized when they, through time, were being adapted to becoming roots, then the roots would be vascularized. So you get both the combination of anchoring the material or the plant in place and allowing for nutrient and water absorption. All right, so that's the roots. Now what happened with the leaves with these plants? Well, the leaves got bigger. Why would leaves get bigger? Because you can photosynthesize more the bigger you are. Not only can you photosynthesize more, you get more surface area, but you can have more stomata. Now remember the thing about stomata are necessary. They're those openings on the bottom of the leaf that let carbon and dioxide in and oxygen out. Again, driving the efficiency of photosynthesis. Now, I'm going to come back to the two types of leaves in a second, but look at the green box over on the right-hand side. About 360 million years ago, the atmospheric carbon dioxide levels began to drop drastically on Earth for reasons that don't have to do necessarily with the plants, but the atmospheric CO2 levels begin to drop. So what natural selection would have favored would have been larger leaves, because larger leaves have more stomata. Now think about it, right? If atmospheric carbon dioxide levels are dropping, in order for plants to survive, and they need carbon dioxide, one of the ways they can compensate is by producing bigger leaves with more stomata, so they can absorb more carbon dioxide. If there's a little bit in the environment, if it's decreasing, more stomata would allow plants to take up more of the little bit of carbon dioxide that is sitting there. However, there's a problem. It always seems that way in biology. It seems that way in life, right? One fix for a problem actually creates another problem. Larger leaves overheat faster. Now, overheating in plants is a little different than overheating in animals, but overheating in any living organism is not good. Overheating causes enzyme denaturation, and enzyme denaturation is going to affect the metabolic activity of the plant. So how do we get rid of the overheating? Well, stomata, if you remember, not only exchange gases, but stomata are also responsible for letting water out. Remember, that was the basis behind C4 and CAM plants. Those that live in dry environments, when they open their stomata, which they have to exchange gases, the problem is they lose water. And I got to remind you back to Bio 1, the C4 and the CAM plants found ways around that. But as far as the overheating part, stomata do a good job of cooling off the leaves through something we call evapotranspiration. Evapotranspiration is plants sweating, basically. That's what it is. Water evaporating off of a surface, and this is a law of physics. Water evaporating off a surface removes heat from that surface. So water evaporating off the surface of leaves is going to cool the leaves down. Okay? All right, now back to the two types of leaves. We have small leaves and we have big leaves. We have microphylls and megaphylls. There's only one type of plant out there today that has microphylls, and they're called the lycophytes. Now we'll get to the lycophytes here in a couple of slides, but lycophytes are the only ones that have microphylls. Microphyll leaves, P-H-Y-L-L, by the way, means leaf. P-H-Y-L-L equals leaf. Okay? So, did I spell that right? Yeah. So microphylls are small leaves with only one vein. Megaphylls are much larger leaves with branched venation. So take down a look down below. This is where we think microphylls may arise. We start with a stem that is vascularized, and then we have an offshoot from that stem, right? And then an offshoot from that stem then becomes vascularized. And then that offshoot that's vascular becomes wider, increased surface area for more photosynthesis. So you can see in the Krause's spike moss, Selaginella kraussiana, there we go, little tiny leaves with one vein, unbranched vascular tissue, small leaves. Now those work, right? Obviously, if they didn't work, those organisms wouldn't be around today. But the typical kind of leaf that you see is the leaf that has branched venation, right? It's got several of those conducting tubes going through it. Here's how we think that may have happened. We get a branching stem with vascular tissue. Initially, the branching is unequal. And I'm glad I have my camera today. What we mean by the branching is unequal, you think about my fingers as being the branches, right? If we looked at it sideways, they would not be in the same plane. I don't know how well you can see that. Does that help? Yeah, there we go. So they're not, you see, we're looking at them, they're not in the same plane. Eventually, through time, they would have arranged themselves through evolutionary changes, through adaptation, through mutations, folks. They would have actually got themselves into a situation where they would have been flat, And then all you got to do, turn back to this way again, is build tissue in between those veins, and you got yourself a leaf. Right? And those are megaphylls, and that's what we typically see in other plants, right? Like you're seeing in that filmy fern there, you can actually see very closely the branched venation that's occurring there. Microphylls would have worked well for smaller plants, right? Megaphylls would have been what we eventually would eventually evolved into all the other leaves we have on plants on the earth now. All right, we talked about leaves, we talked about roots. Let's talk about spores again. Spores are produced on sporophylls. I just told you that phyll means leaf. Everything on a plant, the flowers, everything on a plant is a modified leaf. All right, so a sporophyll is a modified leaf. Specifically, it is a leaf that produces spores. And these sporophylls were a real significant evolutionary trend that occurred. Now, these are leaves bearing sporangia. You take a look at this fern over here. See those dark marks on the back? Those are sori. Eventually, we're not going to get to that today, but when we get into the next chapter, those spores are actually going to be contained within things like cones, if we're talking about cone-bearing plants. And then in flower-bearing plants, those spores are actually going to be retained in the pollen or in the egg of the female plant. Now, the sporangian ferns, we call it a sorus, plural is sori, and in the lycophytes and in most gymnosperms, pine trees are gymnosperms, they form something called a strobilus. A strobilus is a cone. That's actually from the Latin for cone. So you can see down below, these are strobili. I don't know exactly what species this is, but these are cone-shaped sporophylls. All right, now spore production. In most seedless plants, spore production is homosporous. What we mean by that is a sporangium on a sporophyll. Okay, so sporophyll is the leaf. Sporangium, in the case of the ferns, would be the sori. They make one type of spore, and that one type of spore gives rise to a bisexual gametophyte. The fern that we were looking at in lab the other day, on that little fern gametophyte, both the Antheridia and the Archegonia were there, weren't they? Remember I said the best way to tell which is more by location? So that little fern gametophyte had kind of a heart shape to it. And remember I said that the Archegonium are usually found up near the indentation part, and the Antheridia are down here. But they're both on the same plant. They're both on the same structure, I should say. I shouldn't call it a plant. So typically, that's what I mean. That's a bisexual gametophyte. And on that bisexual gametophyte, we have Antheridia and Archegonium. So we're not saying that a bisexual gametophyte has only got Antheridia or Archegonium. We're saying that on that one gametophyte, we have both the Antheridia and the Archegonium. Now, in seed plants, as we move into the next chapter, because we're not going to be talking about seed plants today. 29 is just going to involve plants that are all seedless. So we go from seedless non-vascular to seedless vascular, and then we're going to go seeded vascular plants next. But among seeded plants, what they do is they produce different types of spores. We call that heterospore spore production. So now follow along about what, and I've highlighted in red here. The megasporangium on a megasporophyll produces a megaspore. The megaspore produces a female gametophyte, and that's where the eggs come from. Now, in terms of the male, it's a microsporangium that's produced on a microsporophyll, and they produce microspores. That's the male gametophyte, and that's what produces the sperm. The reason why I highlighted in red the mega, the micro, the female, and the male, the female reproductive cell, the female gamete, the egg, is in most all species that I'm aware of, is usually always bigger than the sperm. So the megaspore, the megasporangium, the mega is the female because that gamete is bigger and the sperm are smaller. Now, if we go back to our cone-bearing plant for just a second up here at the top, these are the male cones right there. That's the male cone. And these are the female cones. So we would call, if we were going to use these terms for that for the male part of that cone-bearing plant, we would say that the microsporophyll is the cone. Okay, yep, these little things, each one of those is a cone. On there are microsporangia that produce microspores that make the female or make the male gametophyte, which contains the sperm. Now the sperm, in the cases of the cone-bearing plants, is going to be transported in pollen. That's what's inside pollen. Inside pollen is the male gamete. On the other hand, the larger cones, which are what we call the ovulate cones, the female cones are much larger. The megasporophyll, remember what I said, a cone is a modified leaf. So a megasporophyll is a modified leaf that makes megaspores, and megaspores again produces the eggs. Okay. Ferns are typically homosporous, and like I said, most all the seeded plants are then heterospores, different kinds of spores. So male and female spores. All right, where are we then? Let's see. So we've gone through these guys, non-vascular, okay, no seeds. We're still in the seedless, but now we're vascular. So we're going to finish talking about these two folks. We'll talk about the lycophytes first, and we'll talk about the manilophytes. Those are the ferns and the horsetails and the whisk ferns. All right, so let's go on down and take a look at the lycophytes. Club mosses, spike mosses, quillworts, those are all lycophytes. I put the word moss in quotes because these are not really mosses. Mosses are bryophytes. These are not bryophytes. These are lycophytes. But the common name, you know how that happens sometimes. Some of these organisms are epiphytes, which means they grow on another plant. I guess the moss, terminology probably comes from the fact of the way they look. These guys were real prominent during the Carboniferous about 300 and 360 million years ago. This is what the Carboniferous might have looked like back then. So you would have had the ferns and lycophytes. Now, lycophytes today are usually very small, like the spike moss. This is a club moss, quillworts. Today they're very small, but back 300 million years ago, lycophytes were as big as trees. These are lycophytes in the background. Horsetails that we'll talk about here in just a second. I always love these kinds of. Look who's still there though, or look who is there. Dragonflies. Dragonflies are so cool. When you look at a dragonfly, you're looking at them pretty much the same way they were back during the dinosaur era, or what we call like a living fossil. I love dragonflies. I find them fascinating creatures. All right, these organisms, like the club mosses, then are homosporous, and then the spike mosses and the quillworts. You can see why the quillworts are called quillworts, because they look like the quill of a porcupine, the thing that jabs you. And they are heterosporous. Lycophytes, and then the manilophytes. The ferns, sometimes it's called the ferns and their allies, ferns, horsetails, and whisk ferns. So we're vascular and still seedless. All right, the phylum Manilophyta. Interesting stuff about this group, the Manilophyta. The manilophytes, even though they're seedless vascular plants, even though they're seedless, okay, they're actually more closely related to seed plants. Take a look right down here. Manilophytes, ferns, and horsetail sinestros shared a common ancestor with seed plants right about there, about what, 380 million years ago? Whereas they shared a common ancestor with the other seedless plants somewhere around 400 and maybe 15 million years ago. So ferns are actually more closely related to seed plants than they are to those other seedless plants that we've been talking about all along. Lady ferns, horsetails, you looked at in the lab, and then a whisk fern. Whisk ferns, I think probably get their name from the fact they look kind of like a whisk broom. Up at the top of the whisk fern in the yellow, these are the sporangia. Oh gosh, there's all kinds of ferns, over 12,000 species. Ferns are probably the most widespread of seedless vascular plants. They can grow relatively tall. This is an Australian tree fern. I had one of those in my backyard until it just got so big I finally had to cut it down. Most ferns are homosporous, so they make a bisexual gametophyte. Some can produce in excess of 1 trillion spores in a lifetime. It's amazing when you think about it. Conspicuous sporophyte. This is the sporophyte. Very small gametophyte. Gametophytes are growing underground. In my little fern garden at home, many times I've tried to very carefully dig down into the dirt to see if I could find the gametophyte. I've never been able to find one. They're very delicate. And since the sporophyte is not dependent upon the gametophyte, since it doesn't grow out of it like in the mosses and needs it for food, then many times the gametophyte just disintegrates after the sporophyte evolves or emerges, okay? Both the sporophyte, though, and the gametophyte are both photosynthetic. Things that are green are typically photosynthetic. Now, I want to point out one thing. The fern in the upper right is a cinnamon fern, and we have cinnamon ferns here in Florida. The reason, is that how you spell cinnamon, or is there 2 M's? Yeah, it's close enough. The reason why they're called cinnamon ferns is cinnamon ferns are a little different than other ferns. I showed you in the back of the resurrection fern, I showed you the sori, right? Go back a little bit. There we go. See the sori? If you were to look at the frond of a cinnamon fern, you would not find sori on it. And you might think, well, now let me preface that by saying that when you look on a fern for the sori, you will not always find sori in the back of the fern. It depends on the season, all right? But when it's the season which spores are going to be produced, on those that produce spores on their fern fronds, you'll easily see them. Cinnamon fern doesn't do that. Cinnamon fern does not produce the spores on its leaves. Cinnamon ferns actually make another structure. No problem, Daniela. Good luck. Good luck on your job interview. This here is the reproductive part. You're welcome. That is actually the spores. So not all ferns do it the same way, right? The spores in the cinnamon ferns and in royal ferns, are close relatives, are actually formed on a different structure. What it basically is it's a modified fern frond. As a matter of fact, if you see a cinnamon fern, if you looked closely at the brown spore part, it almost looks like the green leaves, just brown, and it's all rolled up on itself. It's all curled up on itself. Let's look at fern reproduction then. All right, and we'll start with meiosis again. So here we are, sporangium, sori. There's a sorus. Sorus contain a sporangium. Sporangium through meiosis, variability, produce a lot of spores. Some of them have little springy things that I showed you the other day that helps them disperse. Dispersal is important. So this spore is a homosporous plant. So this is a spore that's going to give rise to a bisexual gametophyte. All right, and that bisexual gametophyte once again has Archegonium antheridia. Archegonium again being found up near the little indentation. Antheridium being found a little bit further down. In the antheridium, sperm are going to be produced. They're flagellated, so they need a little bit of water. Ferns grow in very much the same kind of environments that the mosses do, right? Moist environments. And then if that egg gets fertilized by the sperm, we get a zygote that grows out of the archegonium, and eventually what that's going to do. Now, it's the underside of the gametophyte where the antheridium and the archegonium are found. So this is where the antheridium and the archegonium are. So when that zygote starts growing, what it does is it grows and then it makes a U-turn and grows up through that little indentation. And this little sporophyte, the new sporophyte, is eventually going to become those fronds, the mature sporophyte. Young leaves or young fronds of a fern are called fiddleheads. There's a little bit bigger picture because it kind of looks like the tuning part of a violin. And what will happen is this thing will continue to unroll. So it's rolled up like this. So it's going to unroll, and when it does, as it unrolls, the leaves will then begin to stick out from either side. So that's how these come about. Next slide is the same thing you just see here. Wait, hold on a minute. Let me see something here. Oh, yeah, I wanted to talk just something about bisexual gametophytes. As far as inbreeding, how do they prevent inbreeding? Well, what happens here is that the egg and the sperm are produced at different times. They usually produce the sperm first, the sperm then move their way out in the environment until the eggs are produced later. So that idea is that the sperm have gotten away from the eggs on the same plant, so it minimizes the chances of inbreeding. Doesn't stop it, but it minimizes it. All right, now this is the same picture, just with actual photographs. The fern frond with sori. On the sori are the sporangium. Inside of those are the spores. Those are going to germinate into a gametophyte. All right. That gametophyte, of course, has antheridia and archegonium. And the antheridia are going to produce flagellated sperm. Now, remember in the archegonium, what you're doing is you're looking right down the vase. Down here at the very bottom of the vase is an egg. So that sperm is going to swim from droplet to droplet to droplet until it finally finds an archegonium. It's going to swim down the archegonium. It's going to fertilize that egg. It's going to form a zygote. And growing out of that archegonium, that vase, is going to be that immature sporophyte. That's going to become the leaf of, or the frond of the fern. First it forms a fiddlehead, and then eventually it opens up into a frond itself, a mature frond. Take a look as you're walking around now with the ferns. If you see them, flip them over and see if you can see the sori in the back side of them, assuming that it's not one of those species. And it's only the cinnamon and the royal ferns that are not going to have the sori on the leaves there, around here anyway. Horsetails are another member of this monilophyte group. Horsetails are called that, I think, because they feel like the stiffness of a horse's tail, you know, underneath all the hair. Have a real cool scientific name, Equisetum. You remember the genus of horses was Equus. I don't know if you remember that, but that is the genus for horses. This is called a scouring rush or scouring horsetails. Very diverse 450 million years ago during the Carboniferous. Now we're left with about 15 species, all in one genus, all in one single genus, Equisetum. We have horsetails here in Florida. We have that one, Equisetum hyamale that you see back there, the big one. This is the shoot, the stem that's going to produce the spores, and the other one is a vegetative stem. You can see on the right. This is the vegetative stem. This is the fertile stem. Vegetative stem, those are little leaves, little things that are sticking off. The vegetative stem is going to do the photosynthesis, and the fertile stem is going to be involved in the spore production. All horsetails are homosporous. They're called scouring rushes because when you feel them, they're rough. They have silica impregnated in the tissue. And I think I mentioned in lab, but pioneers used to use them to clean out their pots and pans, kind of like a Brillo pad. That's obviously not why horsetails did that. That's not why horsetails have silica in their tissue so that pioneers could clean their pots and pans. They have silica in there because it's a detriment to herbivores feeding upon them. Grazers, right? Grazers chewing on horsetails would wear down their teeth pretty doggone quick because you're trying to chew up basically powdered glass in a way. So it's a very effective way to discourage predators. I know one place near my house where there's a boatload of horsetails growing. You don't see them very often, but there's one little neat area kind of off in some little secluded area of Oviedo where there's just about 300 feet growing down the side of a road of horsetails. It's really kind of cool. Whisk ferns, there we go. Sporangium are the yellow parts at the top. Whisk ferns are found in the subtropics. I've seen a couple of whisk ferns here in Florida. They're not real common. Living fossils, because they look so much like Cooksonia, right? They look a lot like Cooksonia. Cooksonia was, you remember that first vascular plant that I showed you a couple of slides ago. Let's see if I can find Cooksonia. Keep going. There you go. There's Cooksonia. See, this was the artist rendition of Cooksonia. It looks a lot like a whisk fern, doesn't it? Living fossils are a term we use to refer to organisms that are either have survived since, ancient times. Like, for example, horseshoe crabs here in Florida. Horseshoe crabs, the way they look today, is practically identical to the way they looked 65 million years ago during the dinosaur era. So we would call them a living fossil. Living fossils can also refer to organisms that are around today that look like something from the past. So while whisk ferns are not Cooksonia, right? They're not that ancient plant. They look like Cooksonia. If you're thinking convergent evolution, you're right on the ball. Tropics, no true roots, which we think they may have lost during evolution, growing in the tropics and the subtropics, being that the way the environment is, oftentimes roots aren't completely necessary. Aerial stems without growth. The stem is the photosynthetic part. There are no leaves. So the stem does the photosynthesizing, and then as I said, the yellow is the sporangia at the top. Again, I guess because it looks like a whisk fern. Almost got its name. Or a whisk broom. All right, so here's the world that we got. Back about 450 or so, 425 or so million years ago, we have this expansive growth of these seedless vascular plants. Really huge, tall ferns, lycophytes, mosses, all these things are doing very well. During the Carboniferous period, about 400 million years ago, and the reason why we call it the Carboniferous period is because when we dig down through the fossil record, at about that time in history, we see these huge layers, sediments of carbon, of decaying organic matter. This is actually, if you look down below, it is this material that ends up making coal, because through time and pressure and heat, peat, right, this stuff over here on the left, which is that organic material of plants and everything, gets compressed with heat and pressure and eventually becomes coal. So the Carboniferous period, when you look down through the fossil record, is an area where you see a lot of these coal deposits. Now, during the Carboniferous period, there was an awful lot of forests, okay? The carbon dioxide levels begin to drop, though, and the global temperatures begin to drop. Atmospheric carbon was also being taken up by organisms in the ocean. Calcium carbonate is a very common material in ocean critters. And the calcium carbonate, the CO3, comes from the carbon dioxide. A lot of organisms in the ocean use calcium carbonate to build shells. Like if you think about mollusks, that's what their shells are made up of. So during this time when ocean speciation was occurring, a lot of species were evolving in the ocean, a lot of this atmospheric carbon was going into the ocean. CO2 levels begin to drop, global temperatures begin to drop, and you can see in this chart, yeah, the Carboniferous is about right here. We see these carbon dioxide levels begin to drop off. So this is what the world was, and then as these carbon dioxide levels begin to drop, these seedless vascular plants decreased in prominence. When the carbon dioxide levels begin to drop and the global temperatures begin to drop, it began to become cooler, right? And what we have in a situation there is the environment becomes more diverse and more radical, and other organisms with different adaptations became the ones who kind of went forth from here. And that's what we're going to talk about when we get into the seeded plants. Now there's the calcium carbonate I was trying to show, the shells of the mollusks. Coal, right? 6 million tons of coal is burned every year. Now let me say something, because I've heard it on the news a lot lately. There is no such thing as clean coal. I don't care what anybody tells you. There may be cleaner coal than other coal, but coal is dirty. And coal is a fossil fuel that does major damages to our environment. We have to someday get off of fossil fuels, especially coal. So don't believe anything about someone saying, oh, well, this process of using coal is very clean. It may be cleaner, but it's not clean. So these global temperatures are dropping. The Earth's beginning to dry up at the end of the Carboniferous. The Earth begins to dry up. That means desiccation becomes an issue. Other plants had to find some way to survive. And that's what we'll cut when we get into the next chapter. We'll talk about how the seed plants increased in their dominance after this period. So you see the whole thing I've been talking about in evolution all along this semester. Just when species evolve and adapt to the environment changes. right? Extensive forest during the Carboniferous, lots of bryophytes and lycophytes, doing very well, growing really large, pretty much running the planet. And then the environment changes. Environment changes, dries up, and these plants no longer, especially the ones without vascularization, especially the ones that didn't have the ability to move those nutrients and the water around, especially the ones that had to stay very small and low to the ground like the mosses, right? Because they could just using diffusion and osmosis, they would have been, they would have had a hard time surviving in these much drier climates that came up at the end of the Carboniferous. That's the last slide for that chapter, folks. So I'll finish there today. We'll pick up chapter 30, which is the seeded plants, next week. Do remember you have your lab exam next Monday. And again, to remind you, that's over labs 1 and 4. I'm not really going to quiz you much on lab 3. I'm not going to quiz you on lab 3 at all. I may ask you a few prokaryotic questions, but I'm not going to quiz you on the prokaryotic lab, just the lab 1 and the lab 4. All right, let me stop the recording.